Good morning, everybody. Today is Wednesday, November 18th, 2015. And this is Before the First Cup of Jules here on UCY.tv. Happy Wednesday, everybody. How's everybody doing? It is 52 degrees and partly cloudy here in upstate New York. It actually looks to me, as just looking at the map, like it is warmer in the Northeast than it is down south right now in some locations. So good luck to you on that. I don't know. I'm not looking for it. I saw it looks like by next week we're going to get some polar vortexing going on. So you know what that means. That means the lake effect machine might just turn on. So we're not looking forward to that. But for right now, it's pretty nice and feels like fall. So where do we leave off yesterday? We ended up titling the show Technocratic Hubris and Disruptive Technologies. And we spent the show looking at all kinds of things. By the time I wake up in the morning here, I honestly don't remember what the show was about yesterday. But we did. We looked at all kinds of different things to do with DNA and genes. Honestly, I don't remember. And I heard the show multiple times yesterday for one reason or another for my editing and then the replay. But we did look at disruptive technologies, and that's where we ended up. It included things such as gene editing and, again, I don't even remember. But I'll tell you what surprised me is that I didn't see artificial intelligence and robots in there. That was the one that wasn't in there. Oh, yes, novel new materials such as nanomaterials and whatever. They did have advanced robotics, but I don't know. Did they actually? It mentions advanced robotics, but I remember in that PDF where we counted down 12-1, it mentioning advanced robotics. Regardless, anyway, so we have all these new disruptive technologies that are coming. We talk about them every day. And they pretty much are the things of our nightmares. Okay, so now we have this feedback thing. They are the things of our nightmares, and even more so than anything, I still have to say it is this gene editing like there's no tomorrow. We have CRISPR-Cas9 that we've talked about many, many times, and I actually found a video from Jennifer we'll find her. Jennifer I don't know. Doudna, that was her name, from Berkeley, giving an overview of exactly what CRISPR-Cas9 is. We might look at this, I'm not sure. Well, I mean, we know what it is, but how it works. But what I want to look at here, I found some things that really bothered me yesterday. Gene art strings, DNA fragments, and libraries from Thermo Fisher Scientific. The title of this page is Synthetic Genes Ready to Clone, Affordable for Every Lab. Now, you know how we talked about Johnny having a little lab kit now? They've been saying this, look out for terrorists who will be genetic engineering in their garage. But little Johnny has a lab kit where he can splice and dice DNA. And now he can go online and he can order it. Mail order. Gene art strings, DNA fragments, and libraries are now available with sequence fidelity using full, which who even knows what these are, IUPAC code of degenerate nucleotides. Just what I want in my cloned lab Frankenstein. Degenerate nucleotides. It says gene art string DNA fragments are custom made, uncloned, double-stranded linear DNA fragments from 150 base pairs to 3,000 BPs in length, assembled from synthetic oligonucleotides using the same high-quality process developed for gene synthesis. Strings DNA fragments are delivered dried, ready for resuspension, cloning, and screening to enable the identification of the correct clone. Strings DNA fragments are fast and small alternative to get your synthetic gene and clone into your expression plasmid. What is this? I mean, this is an ad, people. They're selling synthetic gene fragments via mail order to whoever wants them to use for your gene and clone expression in your plasmid. I don't even know what that means. At least 200 nanograms of strings DNA fragments are produced within 5 for up to 1 kilobyte, or 8 for 1 to 3 kilobytes business days. So I guess they can make them for you quickly. Gene art string DNA libraries are pools of gene art string DNA fragments from 200 to 2,000 BP containing up to 3 blocks of degenerate nucleotides with randomized distribution. See the schematic below. Each block can consist of up to 300 BP using full IUPAC code of DNA nucleotides. Blocks need not be separated. I don't even know what this means. You can get them all within 10 to 15 business days. They are affordable, cost-effective alternative to gene art gene synthesis. They are flexible. Full gene design and cloning flexibility. Enter your sequence and directly edit, optimize, and order through our online portal. And they're fast. Production time starts at 5 business days. So this looks like you can literally go in, design, and order custom DNA sequences. I don't know. I mean, I kind of joked about this stuff. I remember seeing some mail order DNA the one time, and I was like, wow, that's kind of weird. And now we're at this level. We have online design portals for synthetic DNA that then they'll make for you. Well, yeah, it gets even better. Gets even better. Here's the do-it-yourself CRISPR genome editing kit that brings genetic engineering to your kitchen bench. November 11th, 2015, out of Gizmag. CRISPR genome editing is one of, so again, disruptive technologies. Like, this is beyond disruptive. This is end of the world type technologies. I don't even know why they're letting people play with this stuff. This should all be highly, tightly controlled. And I don't even think we should have this ability, honestly. This bugs me. CRISPR genome editing is one of the most significant world-changing technologies of our era. No kidding. Allowing scientists to make incredibly precise cut and paste edits to the DNA of living organisms. Now, one synthetic biologist from NASA plans to make it as accessible as a home science kit so you can biohack yeast and bacteria in your kitchen. And yeah, again, remember those biohackers we talked about? People doing things. I mean, I've always been one to use myself as the guinea pig first and try experiments, but I was never like cutting myself, stuffing LEDs under my skin. That was the newest one we just saw about biohackers. Um, so now you can hack bio yeast and bacteria on your kitchen bench. If you are not up to date with how CRISPR gene editing works, take a short look at this excellent MIT video, and I think that is the one of Jennifer Doudna, who's the creator of CRISPR Technologies. I don't know. Do we do that? It's been a really long time since we have heard a video. I mean, like, really long. So let's see if I can remember how to do this. I don't know. We'll see if it works. Maybe we'll get lucky. Or maybe we won't. I don't know. It's not even calling me. Oh, here we go. Maybe. Can you hear me? I think so. I think it's all working. All right, let's see if this works. This would be Jennifer Doudna. And what is CRISPR Cas9 technology and how it works. And I think this is important for all of us to understand here. All right. Here we go. Yeah, it's proteins. Proteins to cut your DNA.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA.
Okay, what a crappy piece of shit machine this is. I'm going to dump this sucker and we're going to move over here. I don't know though, this machine, at least this one works, but this one is where everything is open. So hold on. God, this is frustrating. This is why I just gave up playing videos, because it totally ruins my flow and pisses me off and then makes me not even want to be here. Well, this might not fucking work. Wow, is this irritating? Why does everything have to be such a piece of shit? It's like everything is a piece of shit. Oh. I mean, wasn't there a day when stuff just worked? Because I thought so. At one point. All right, let's try this. It's like every machine has an issue. Even though these are all better now. All right, here we go. Let's try connecting again. Jesus. All right. Oh. Can't have speakers plugged in. Wow, that was some awesome feedback. All right, Jennifer Doudna, UC Berkeley, MIT, genome engineering with CRISPR-Cas9. Let's hope this one works.
Hi, my name is Jennifer Doudna from UC Berkeley, and I'm here today to tell you about how we uncovered a new genome engineering technology. This story starts with a bacterial immune system. That means understanding how bacteria fight off a viral infection. It turns out that a lot of bacteria have in their chromosome, which is what you're looking at here, a sequence of repeats shown in these black diamonds that are interspaced with sequences that are derived from viruses. And these had been noticed by microbiologists who were sequencing bacterial genomes, but nobody knew what the function of these sequences might be until it was noticed that they tend to also occur with a series of genes that are often encode proteins that have homology to enzymes that do interesting things like DNA repair. So it was a hypothesis that this system, which came to be called CRISPR, which is an acronym for this type of repetitive locus, that these CRISPR systems could actually be an acquired immune system in bacteria that might allow sequences to be integrated from viruses and then somehow used later to protect the cell from an infection with that same virus. So this was an interesting hypothesis, and we got involved in studying this in the mid-2000s, right after the publication of 3 papers that pointed out the incorporation of viral sequences into these genomic loci. And so what emerged over the next several years was that, in fact, these CRISPR systems really are acquired immune systems in bacteria. So until this point, no one knew that bacteria could actually have a way to adapt to viruses that get into the cell. But this is a way that they do it. And it involves detecting foreign DNA that gets injected, like shown in this example, from a virus that gets into the cell. The CRISPR system allows integration of short pieces of those viral DNA molecules into the CRISPR locus. And then in the second step of that's shown here as CRISPR RNA biogenesis, these CRISPR sequences are actually transcribed in the cell into pieces of RNA that are subsequently used together with proteins encoded by the Cas genes, these CRISPR-associated genes, to form interfering or interference complexes that can use the information in the form of these RNA molecules to base pair with matching sequences in viral DNA. So very...
Oh my God. That made me even more confused, but I heard a couple keywords. They use interferences for RNA. Well, that's RNAi, being able to flip switches off. It also sounds like what they're doing is, so the bacteria, they found this immune system in bacteria that fights off viruses when viruses get in and start replicating. So I think what they're doing is they're letting, because they suck the guts out of the viruses and then they put new guts in. I don't know. We'll hear more. I think I really need to take a genetics class. This is pretty mind-bending, especially first thing in the morning. And I am only like a quarter of the cup way through my coffee.
A nifty way that bacteria have come up with to take their invaders and turn the sequence information against them. So in my own laboratory, we have been very interested for a long time in understanding how RNA molecules are used to help cells to figure out how to regulate the expression of proteins from the genome. And so this seemed like also a very interesting example of this. And so we started studying the basic molecular mechanisms by which this pathway operates. And in 2011, I went to a scientific conference and I met a colleague of mine, Emmanuel Charpentier, who is shown in this picture on the far left. And Emmanuel's lab works on microbiology problems, and they're particularly interested in bacteria that are human pathogens. She was studying an organism called Streptococcus pyogenes, which is a bacteria.
I'm going to stop here because you know what? You know what it feels like Monday, but this isn't even the same video that we had queued up on the other machine. Come on. Hold on. This is a 16-minute video because I was like, why are we sitting here talking about this other guy? Hold on. See, today was one of those mornings I really felt like staying in bed. I don't know. I'm having real difficulty here. All right, here we go. It's all right, guys. I don't have one of these too often. But see, this is what happens every time I do a clip lately. That's why I don't even bother doing them anymore, because it's just such a pain. Such a
match it to its target.
All right, stop, because we are not that far. All right, here, we're going to start all over again, but this is totally different. You know what? That was so confusing. That was Jennifer Doudna, but she was given some sort of lecture at the microbiologist level, and I'm just not there. So here.
Consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus.
I got it. So this is literally what we've learned to do is to hijack the immune system of the bacteria that's protecting itself from a viral invader. All right, let's hear a little more. All right, I think maybe I can figure this out.
Over the past few years, researchers studying the system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target.
Oh, this is scary. So here's what they're doing. So they're taking this immune system of the bacteria that goes in then, and when it recognizes an invader and its original case, this viral DNA, it goes in and it matches with this RNA strand that it makes. It matches what it has there in the viral DNA. It makes a duplicate and it zips them together. I don't know if you've ever seen the way RNA can attach and recombine, but it's like a zipper where they match sequences and then zoop and pull all together into a full sequence. So what these guys are doing now is that they discovered that they can make matches for any type of DNA that they want to remove. So if they want to remove genes that are, what were we just reading about the other day that was supposedly really simple, some sort of, or maybe they were thinking about it, some sort of genetic eye disease we were reading about yesterday because it was localized in the eye. They were going to give that a shot where they'll be able to match that sequence, make that zipper piece inside the CRISPR gene or whatever they're calling it. And then when they let that loose, that's going to treat the eye disease genes that they've matched now as a foreign invader, so the immune system of this bacteria goes in and cuts it out. So they're using the immune system of this bacterial cell as their gene executioner. All right, I think I'm getting this now. All right, let's hear a little bit more.
And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA.
Oh my God! So we really want something that they're going to inject inside of us that can start unzipping our DNA zippers? I mean, what happens when your DNA starts unzipping? I think you turn into a molten pile of gelatinous biological material. This can't be good. What happens if this went uncontrolled?
If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version.
So once they get it to mutate, they cut it, it tries to repair, it doesn't quite repair itself properly, they can then go in and insert their own version of whatever DNA sequence they want to put in there and get it to repair itself with that.
All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together.
Well, that's the one thing that we've talked about before, and we saw all this when we looked at some of the transgenic mosquitoes that they've been making, that they literally, this is what's so dangerous about this gene drive, the CRISPR gene drive is what they're calling it, because once they start one of these processes, this can replicate throughout an entire living organism. I mean, they can go through and take some genetic defect they've decided you have that they want to correct, and they could spin that up and have it spread all throughout your body in the name of a cure. I mean, no thank you. They have no idea how much damage or insanity could ensue with this.
These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
That's McGovern Institute for Brain Research at MIT. You know, the scary thing is did you hear her say too that it's for medicine and whatever, and for agriculture? This is the same sort of stuff that they are doing now with the new GMO foods, this next generation, and it's using, and they, you know what, this wasn't very good because it still didn't explain to us like how the RNA works and how it interferes and what it does. So I guess we're going to have to dig more into this. They're also using the same technology for drug development and all this other stuff. And I see here's another CRISPR technology video from AstraZeneca, who of course is a large pharmaceutical manufacturer. But I don't know. Well, I guess we're going to have to I really don't want to like do a whole lesson into CRISPR, though. Now's the time. I mean, this show's been kind of already lame-o. So I don't know. Let's see. I really wanted to. I had a couple really good stories today that I don't think are going to happen. All right, let's see. Oh.
Targeted genome modification has been hampered by the lack of an efficient tool to create double-stranded break at designed location. The CRISPR-Cas system is an RNA-guided nuclease system for targeted introduction of double-stranded DNA cleavage. Originally discovered in bacteria as an acquired defense against foreign pathogen, CRISPR-Cas technology has been demonstrated to work successfully in all the common model organisms, making it one of the hottest technology breakthroughs in 2013.
Here we go. One of the hottest technology breakthroughs. And you remember, over the last, I don't know, few weeks, we've been looking at all this crazy stuff with precision medicine and the tech titans wanted to live forever, we've discovered that this is all being done for a buck. This is the new economy. This is the way it goes. It's you, me, all of nature, old people, of course. We have the entire planet getting older as reproduction drops off. Countries such as China and Japan that are having a real growth in the older generations, and that is just prime pickings for any of this technology. And of course, we have model organisms. They have mice and salmon and worms and bacteria. And let's just try to hack the entire microcode.
Targeted genomic cleavage requires two key elements, a homing device and an endonuclease. In the CRISPR-Cas platform, the homing device is guide RNA, or gRNA, and Cas9 is the enzyme introducing the double-stranded cleavage. The gRNA contains a 20-nucleotide sequence linked to a tracer RNA scaffold. The targeting sequence is immediately upstream of a protospacer-adjacent motif, or PAM. This is sufficient to direct the Cas9 nuclease to the complementary site in the genome and create double-stranded break. Compared to previous genome editing methods, such as zinc finger and TALEN, CRISPR-Cas is cheaper, quicker, and more accessible for researchers.
Yeah, I think we're going to have to leave it at that because this is like mind-bending right now. I need to find a better, some like 101 kindergarten CRISPR-Cas9, hold my hand and teach me so then I can digest it and regurgitate it, because I'm still really confused. And I mean, I dig into this stuff and I just still don't get exactly how this is working. So I don't know. My hope was that somebody would have been able to explain it very simply, but I guess not. So here we go, though. It gets even worse than all of this going on. And this is something I wanted to share. I ran across this after the show yesterday, but I've been looking for the story for probably the last, I'd say 11 months, considering the story came out January 3rd. I remember posting this when it came out, and I even mentioned it, and then I went back a few days later to find it, and it was completely gone. I could not find it. I couldn't search it. I couldn't remember even what the keywords were in the title, and it just kind of slipped through the cracks. Well, hey, I ran across it again. I think we're just going to read. I'm not even going to give you the title. Oh, maybe I should. I ran across the name of a company a few days ago when we did the show. Maybe it was Friday, maybe it was Monday. And I wasn't here Monday, so it must have been Friday. The name of the company was Cambrian Genomics, I believe. And I dropped their link in the chat. I had it open here on my desktop, and as I was cleaning things up after doing my notes, I went out to that link. I clicked on Cambrian Genomics, and go out there. You could take a look. And I was like, what a strange website. It's just like a shell, like a pre-Cambrian shell, like it looks like a mollusk or something, prehistoric mollusk. And I don't even have it in front of me to see what it looks like, but it's got like this little laser beam, and it says Cambrian Genomics DNA laser printing. It's like, what? And there were no links to anything. There's an address to contact them, and that's it. That's all it has. So I was like, whoa, what is this? Why is there nothing on this website? They came up as like one of the darlings in the story. So I did a couple searches for Cambrian Genomics, and I was like, oh my God, here is the story from January that I couldn't find that I wanted to share with you guys, and it disappeared. Controversial DNA startup wants to let customers create creatures. Yay! So this gets even better than mail order DNA. It gets better than plug it in online and we'll have it for you in 8 days. Oh, yes, it gets way, way better. In Austin Heinz's vision of the future, Customers tinker with the genetic codes of plants and animals and even design new creatures on a computer. Then his startup, Cambrian Genomics, prints that DNA quickly, accurately, and cheaply. Anyone in the world who has a few dollars can make a creature, and that changes the game, said Heinz. That creates a whole new world. What? Anyone with a few dollars can make a creature. That absolutely creates a whole new world of absolute, utter insanity. What on earth? Why isn't somebody taking people like this and saying, no, we are not creating hybrid, not even hybrid, using your imagination to create life forms, mail order, with laser beam printing of DNA sequences. I mean, why? I cannot understand how this is even possible. Where the hell is regulatory agencies on a global level? Guys, global. And you know I'm not for globalization, but I'm telling you, this needs to be locked down in a vault. Nobody has it. Not DARPA, nobody. Nobody. Danger Will Robinson. This is it. This is hacking everything that exists right now. The 31-year-old CEO has a deadpan demeanor that can be hard to read, but he is not kidding. In a makeshift laboratory in San Francisco, his synthetic biology company uses lasers to create custom DNA for major pharmaceutical companies. Its mission to, quote, democratize creation, unquote, with minimal to no regulation, frightens bioethicists as deeply as it thrills Silicon Valley venture capitalists. Yes, remember all those billionaire tech titans that are pouring all their money into this stuff? Yes, those same Silicon Valley venture capitalists that are so damn excited. Now, if we could be walking around passing laws about how you're going to inject me and my kid with this shit, forcing it on us without any type of proper informed consent, putting it in my food without telling me, it screws with our immune systems and makes us sick from every which way to sideways, incorporating all this shit into our bodies and making us guinea pigs. You can do all that, but you can't even reel in like pure old school capitalism for gene editing and creature creation. There's a big problem here. The latest technology and generous funding and a growing number of startups are taking science and medicine to the edge of science fiction. Yeah, well, shouldn't that be a problem? Like, I can't even get over how retarded this person is writing the story without any ounce of concern, like what this means. Oh, yes, look, we're going to have flowers that can change color and milk made without cows. Oh, it's so wonderful. Animal-free meat, that's what they said. It's in the works or on the market are color-changing flowers, cow-free milk, animal-free meat. Test to detect diseases from one drop of blood. And actually, yeah, I have that story queued up too. And pills that tell doctors whether or not Nana's taking her medicine. Wow, I'm so excited for the future. Totally new organisms. But few founders are pushing the technical and ethical boundaries of science as far as Heinz, who told the Wall Street Journal, quote, I can't believe that after 10 or 20 years, people will not design their children digitally. There you go. There you go. He said it, and he's a guy in the know for certain. We knew that that was going to be possible. And it's pushing toward that. Anything for a buck. You're going to be able to create your own X-Men. And guess what, though? It's not going to be any type of natural fertilization, and you're going to have to be chosen to procreate. You know, it's not going to be any of us defective genes that are procreating. Any of us that have had too much stress or kind of taken life a little hard, done damage to our DNA stock. In a recent conference in Vienna, Heinz said, quote, "We want to make totally new organisms that have never existed." Now, it'd be very interesting to see this conference in Vienna. Cambrian Genomics CEO will design every human on a computer and make poop smell like bananas. Oh my God. Say this for Austin Heinz. His vision for the future will either thrill you or leave you fearing for the future of humanity. See, now we did a story yesterday about people with a sense of humor. Beware of people with a twisted sense of humor because they could have dementia. I think this dude needs to have his head checked. I think anyone in charge who's allowing this to continue unchecked needs to have their head checked. Yes, leave you fearing for the future of humanity. And not just humanity, for the entire biological biome that exists on this planet. Anything can be sliced and diced with this. And it even says, either thrill you or leave you fearing for the future of humanity. There's not really any room in the middle. In a pair of interviews off and on the stage at the Pioneers Festival in Vienna today, and this was back in October of last year, the CEO of San Francisco-based Cambrian Genomics explained the mission of his company, which is often benignly described as, quote, laser printing DNA. But what does that mean? Says, we want to make everything that is alive on the planet, he explained. Everything that is alive is not optimal. It can be made better. Ding, 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 ding. We've talked about that as well. And once they've bag tagged, cross-referenced, sliced, diced, and put us all in a database, and they understand what all these different genetic modification or genetic traits apply to, and what's considered a good trait and what's considered a bad trait. You know, don't think that there's going to be those of us who are deemed insufficient stock, who are deemed defective humans. Defective humans for so many reasons. And I mean, nobody's perfect, right? That's what they always told us. Nobody's perfect, Jenny. You know, we all make mistakes. Try better next time. Yeah, well, you know what? You can't change your genes. And we even saw something the other day where, and I'm trying to remember what the story was about. They were looking at crime and people with genetics that are related to someone with crime. And I don't know if this was hypothetical or if they were really doing it, but they have actually found genetic traits that seem to be passed down that are responsible for deviant behavior. And what would that mean? Like if you had somebody who committed a crime in your family 5 generations back, does that mean that they're going to have to gene edit that out of you, or that you would have never even been born because they determined that anyone with a criminal history or a criminal gene or a defiant gene doesn't get to reproduce? Or if they do get to reproduce, that is a mandatory gene edit that needs to happen to the embryo before it's ready for implantation. This is really, really unbelievably horrifying, the implications that we're talking about here. And everyone's just la-di-da-di-da-di-da. Oh, this CRISPR technology is going to make us bazillions of dollars. Let's just keep watching TV. I mean, that's what it is. People do not understand that we are at the level and we're giving this power to lunatics like this. Everything alive is not optimal. It can be made better. So let's give him the keys to start tinkering with making life better. Anyway, back to the story. He doesn't have plans for replication. Quote, we want to make totally new organisms that have never existed, he said. So this was talking about like the bipedal wormhead yesterday. Quote, we want to make totally new organisms that have never existed, he said, and replace every existing organism with a better one. It just seems obvious that eventually every human will be designed on a computer, unquote. Now, this is the second echo that we have heard now. First time was from the DARPA lady, and now from this, I don't even remember his name, Heinz, Cambrian Genomics CEO. We're going to replace everything. We can make it all better. You just need to be a non-resistant consumer. Take your vaccines and eat your GMO. Shh, we'll take care of it. Lest you think these are ravings of a mad scientist or the opening scene of a new sci-fi dystopian thriller, well, it's not. Heinz is calm and rational in his view, and while his company has not disclosed its total fundraising, it's backed by such notable Silicon Valley names as, hey, Peter Thiel again rears his ugly head. And they're going to design life forms, guys, like real ones. The technology at work here is complex, but essentially Cambrian Genomics says that it has found a way to dramatically reduce the cost of printing a strand of DNA. Sequencing of the genome is so advanced, Heinz explains, that basically people can take DNA code from libraries and create organic mashups, much the way a programmer can pull computer code from code libraries. And that new code is then laser printed for a fraction of what it used to cost. The most notable example of this technology, we talked about this the other day at work, was the Glowing Plants campaign on Kickstarter. Using these synthetic biology techniques, the campaign promised to take bioluminescent genes from bacteria and fireflies and insert them into several plants to make them grow in the dark. And we saw that the other day with the glow in the dark trees. It's something that they're tinkering with in the Netherlands. The backers raised $483,013 from 8,433 idiots. Why would you want to start splicing and dicing trees and plants just so you could see them glow in the dark? Leave well enough alone, people. Enjoy the beauty that we have in front of us. Don't bastardize it in your own psychotic image. There is so much that is wrong with this. I mean, you know, when you start looking at ethics and the ethics of life and what is ethical and what is not, I mean, I don't know. I kind of think I have some sort of sense for right and wrong. You just got to follow that internal moral compass. This is wrong. What gives us the right to do this to another organism? Did the tree say that it wanted to be glow in the dark? Because I didn't hear him. This is like after Fukushima. We saw them. They started messing around with that bioluminescent gene, and they were splicing it into like goldfish, and that was the newest rage in Japan. It's glow-in-the-dark goldfish. Like, were they trying to make people feel comfortable with glow-in-the-dark radioactive? What? I don't understand what they was about, but glow-in-the-dark goldfish were all the rage there for a while. Now it's going to be glow-in-the-dark trees. Soon to be glow-in-the-dark kids so you can find them when the lights are out. Anyway, now Heinz is fighting back. He says on stage that the company will soon launch its own crowdfunding platform for GMOs called Creature Creators. Yay! Cambrian will also help other people create slick videos to promote their synthetic biological creations. So after you create one at his company, then he's going to help you make a promo so you can sell it or something. One such project he mentioned on stage was Petomics. Oh my God. The project essentially... Oh, all right. So this isn't like genetically modifying pets. The project is essentially aiming to take the genetic material for the odor of bananas and inject it into E. coli bacteria. Oh, God. The modified bacteria would then be introduced into Fido's food. Yes, your pet food. And the result would be pet poop that smells like bananas. Oh, my God. And Heinz says, it's a pretty strong smell, too. Oh. Heinz doesn't appear to fear the potential backlash such biological tinkering could provoke. For him, there is a simple logic at work that makes such technology inevitable. Just give it up, embrace it. It's inevitable, says Kurzweil. People are essentially badly designed computers, he said. We are running a program that is designed for us to die, he said. And I think obviously every organism will be designed synthetically. It's not a stretch to take it to every human and everything that's alive. I mean, but wait a second, Heinz. Who says that the cycle of life, death, and rebirth is wrong? Why is that defective? It's beautiful, actually. Everything recycles itself naturally. If everything lived forever and it was non-biodegradable, we went from carbon to silicone, or whatever the hell you're using, from analog to digital, we've lost the connection that connects us to the planet, the life cycles, the universe. Everything works in these cycles, the life, death, rebirth. That's how nature works. That's the system that we are within. So by thinking that we could just defy death, make everything alive, nothing dies now, because we're defective, so we're going to fix it. That's really psychotic thinking. And that's all of these tech titans on these longevity projects. Their goal is immortality. It isn't going to be you or me, defective Johnny, living forever. Absolutely not. This is going to be in the hands of the few billionaires who can then take their wealth and pass it on for thousands of years as they sit in charge. I mean, again, hubris. And these people don't realize, or maybe they do, but they're just painting it as, well, it's so inevitable, we might as well just let it happen. Shh, close your eyes and eat the banana. I don't know. I don't even know what to say about this. All right, we're going to bounce back to this other story that I was reading, because then I jumped out and went to that Vienna conference. So we want to make totally new organisms that have never existed. His 11-person team has raised $10 million from more than 120 investors, including Peter Thiel's venture firm Founding Fund, or Founders Fund. It is a fundamentally new technology that can open up a whole new industry, says partner Scott Nolan. Venture capitalist Timothy Draper, another investor, praises Heinz as an, quote, exceptional leader with a unique passion for his business. I love Cambrian, he said. The company is literally printing life. Oh my goodness. Can't wait to see all the great things that come of it. I mean, what? To be clear, Cambrian isn't printing designer babies or dinosaurs yet. Still, his rhetoric alarms critics. Marcy Donofsky, executive director of the Center for Genetics and Society, a bioethics watch group at Berkeley, sums up Heinz's belief that, quote, every problem can be solved by engineering is a kind of techno-libertarianism. We have to take seriously people like Austin Heinz who say they want to modify future generations of human beings and upgrade the human species, she said. I think that technical project is far more complicated than they acknowledge. Well, yes, but nonetheless, their story about what we should be striving for as human beings and as a society, I think, is very troubling. Powerful technology, and it gets into the ATCG and how this all works. The DNA printer technology could be used for more superficial means like printing desired eye and hair colors. And Heinz has no problem with that. People are already trying to make those decisions by deciding who they're going to breed with. Well, I don't know about that. And then there's the potential matter of creating living things that are now stuff of science fiction. If you could take a chicken and make it the size of my building, you would probably learn a lot about genetics, which could be useful for human applications. So let's make Godzilla chickens. Oh, my God. All right, guys, well, make sure you stick around because I think it's Wednesday. Yes, it's Wednesday. So Ms. Lonnie Clark is coming up next. And then at 6 p.m. Eastern, we have Josh with Anti-Tyranny News live. The Boys from New York at 8. Truth or Talk with April and Verstein at 9. And Anti-Media with Nicky DB at 11. All right, guys, everyone take care. Have an awesome Wednesday.